The effect of visible light in different sodium chloride concentration on seed germination in plants.
Hi, my name is PRADIPTA NARARYA WARDHANA. I'm an active student cadet in military biology program at the Republic of Indonesia Defense University. In this video, I'd like to experiment with finalist. You can call them among bits. This experiment titled The Effect of Visible Light and Different Sodium Chloride Concentration on Seed Germination in Plants.
Goals.
This experiment goals are to determine and analyze the effect of visible light on finalist seeds germination, and then to determine and analyze how the salt solutions with various concentrations will influence on finalist seeds germination, and then to determine growth velocity of finalist in various treatments.
Seed germination. Seed germination is a process of a seed develops into a new plant. This process is initiated when the seed absorbs water, called imbibition. Absorbing water causes the breakdown of stored nutrients that triggers early growth. There are several environmental factors that could influence the germination, including water availability, temperature, oxygen, and light. Different species have specific germination requirements and conditions such as salinity, pH, or chemical inhibitors. Tools and materials. There are plastic cups, finalist seeds, cartons, water, sodium chloride solids, ruler, analytical balance, beaker glass, spatula, and bottle cap. Variables.
In this experiment, there are three variables. The first variable is control variable. That are seed type, number of seed, watering volume, and planting media. And then the second variable is dependent variable. The dependent variable divided into number of seed that germinate and sprout length. And then the third variable is independent variables that are concentration of watering solutions and light provision.
Treatments.
In this experiment, I treat the mung beans into 6 treatments that are P1, P2, P3, T1, T2, and T3. The first is P1. The P1 treatment is a treatment that treat the mung bean into the bright place and watering the mung bean with 0 molar sodium chloride solution. And then the second treatment is P2, that is treat the mung bean into the bright place and watering the mung bean with 0.05 molar sodium chloride solution. And then P3 treat the mung bean into the bright place and watering the mung bean with 0.1 molar sodium chloride solution. And then the fourth treatment is T1. T1 treat the mung bean in the dark place and watering it with 0 molar sodium chloride solution. And T2 treat the mung bean into the dark place and watering it with 0.05 molar sodium chloride solution. And then the last treatment is T3 that treat the mung bean in the dark place and watering it with 0.1 molar sodium chloride solution.
Procedures.
Making salt solution. Firstly, calculate the sodium chloride solids masses to make 0.05 molar and 0.1 molar sodium chloride solution. And then mix 1.75 gram of sodium chloride solids with 600 milliliter waters in the glass beaker to make 0.05 molar sodium chloride solution. Finally, mix 3.5 gram of sodium chloride solids with 600 milliliter waters in the glass beaker to make 0.1 molar sodium chloride solution.
And then observing Vigna radiata. Firstly, soak Vigna radiata seeds in a bigger glass. Then, if they have been soaking for a night, choose seeds that drown in the cups, then dry them off. Thirdly, put thick cartons into each of the thick plastic cups. Fourthly, put 4 Vigna radiata seeds into each cup, then marking cups according to the treatments that will be given. Here are the given codes of the treatment: B1, B2, B3, D1, D2, and D3. And then watering cups with suitable solution according to the given codes. Use bottle cap as a measurement. And then placing cups with code B in a bright place and D cups in a dark place. Data and discussions. The experiment results show that visible light has a negative effect on the stem length growth of Vigna radiata. It can be seen in Table 1 and Table 2 that sprouts placed in the dark coded D1, D2, and D3 have higher stem length and growth rate than the sprouts exposed to light that coded with B1, B2, and B3. However, the stems of sprouts exposed to light were greener, fresher, and stronger than those not exposed to light. This is because the activity of the hormone auxin in the stem of the sprouts is inhibited by light, resulting in slower stem growth. However, the presence of light causes chlorophyll in the sprouts to work optimally so that the stems become greener and stronger. Salt concentration negatively affects the germination and growth of Vigna radiata. Table 2 shows that sprouts watered with 0.1 molar sodium chloride that coded with B3 and D3 grew the slowest compared to the other samples, while the sprouts watered with 0.05 molar sodium chloride that coded with P2 and T2 grew slightly faster than the 0.1 molar sodium chloride treatment, but not faster than the 0 molar sodium chloride treatment that coded with B1 and T1. In addition, it can also be seen in the Table 1 that the stem length of samples B3 and D3 is the shortest than the other samples. The cause of the inhibition of Vigna radiata growth by salinity is because the high concentration of sodium chloride causes high osmotic pressure in the growing medium. Thus, the roots have difficulty in absorbing water. In addition, salt levels cause oxidative stress in plants, which results in disruption of photosynthesis and metabolism of Vigna radiata sprouts. Then, the ionization of ions from sodium chloride compounds have the potential to be toxic, which will inhibit the growth of Vigna radiata. The conclusions. The experiment proves that visible light and sodium chloride concentration affect the germination and growth of Vigna radiata sprouts. Visible light inhibited the process of stem length increasing of Vigna radiata. This is proof from the difference in stem length and growth rate between the light and dark conditions. The speed of stem growth in dark condition tends to be higher than in condition exposed to light. Even so, the resulting stems have a greener, fresher, and stronger stem. Besides that, salinity only inhibits the stem length increasing of Vigna radiata. The evidence is that the growth of Vigna radiata stems in 0.05 molar sodium chloride treatment is faster and produce longer stems than samples treated with 0.1 molar sodium chloride regardless of dark or light conditions.